All right. And so we left off on our sarcomere that it was made out of thick and thin filaments. And on our packets here, noted by this purple one, is going to be our thick filament. And we gave a name to these thin and thick filament arrangements. The unit is called a sarcomere, and this just keeps repeating itself. If we see the little other piece of our handout, you can see that here's the beginning of one sarcomere, and here's the other till the very end of that same sarcomere. But this unit repeats itself. So this is what I'm going to write on this. It starts here, and then the next one here. So what we're seeing is 2 total sarcomeres at the moment, just highlighted there. And it has names or terminology around the arrangement or the organization around those thin and thick filaments. And so when we look at the center thick filament, it's considered our M line, because the M line, you can even think about it like mid. And so it sits in the middle of a sarcomere, and here we're seeing our bracket indicating our sarcomere. The boundaries of our sarcomere are the Z lines, and I like to use the word like you're zigzagging in traffic, right? Zigzag, like to scramble left and right as you're trying to dodge. I don't know, maybe someone's throwing something at you or something, dodgeball. And so Z lines sort of is the ones that you see here that make little Z's there. And so that marks the boundaries of a sarcomere. Then we have our A band, which is defined as the entire length taken up by our thick filament. There is overlap with thin filaments, but it's something we do not care about when we refer to the A band. It's really from thick filament to the end of that thick filament, that range, and that's why you're seeing this bracket. And then we have our I band, which marks the boundaries of where you have only pure thin filaments. As you can see here, you really only have thin filaments, and it lacks thick filaments. And so, within this space, around our M line, you have in the center region here your H band. Your H band is what changes when a sarcomere contracts. And if we had to speak very simply about this, is what we're talking about in general, like what we're learning is something called the sliding filament theory, which means that this is what we believe that these myofibrils are doing in order to go from being a longer muscle, right, being fully extended, to contracting and decreasing in size. And to really understand it simply, it goes from Z line to Z line. These move towards the M line, or these thin filaments within this boundary aim for that M line, so that during a contraction, that's the movement. And that's the real relevance of our H band. That our H band is denoting the distance between one thin filament versus the other thin filament, and this H band decreases in length as we contract. And all you have when you really think about a sarcomere is you have this accordion-like structure that they're all decreasing towards this M line, and if they are all decreasing in this manner, then you can expect that our length will decrease overall. And this is the sliding filament theory. And so, understanding the anatomy of a sarcomere becomes essential. You must learn A band, I band, Z line, H band, zone of overlap, and Z line. And yeah, we'll just leave it at that. And so, when we examine thick and thin filaments alone, myosin was the main molecule, the protein molecule that makes up this myosin. And you can see that this is just a single myosin molecule, but if you bound a bunch of them together, this is what you can expect, is that you expect this is the tail end, this is the head end, and they're just all bundled up, and that's what thick filament is. You can even see here's their center with our M line, and that would be, again, corresponding to the purple ones that we see on our handouts. Our thin filaments, which are these red, pinkish ones, when you look at thin filaments, you can see that they're slightly different, and it's composed of F-actin, nebulin, composed of tropomyosin and troponin. Now, F-actin is the name of the entire molecule itself, and nebulin, what it does is that it keeps all of these actin molecules anchored together. So I'm going to repeat this one more time. Our nebulin anchors all of our G-actins. But together, the nebulin and the G-actin, with some new molecules that we're going to learn about, the tropomyosin and troponin, which will be essential in learning soon, they make up our F-actin. So F-actin is made out of everything you're seeing here. Okay, everything you're seeing here. And G-actin are going to be the spherical-like ones that contain our active sites. So let's write that in there, contain active sites. Let me put this down somewhere more comfortable for me to, right? Yes, our nebulin holds our G-actin together. Nebulin holds our G-actin together. And our G-actin, where it'll be most relevant for us, is it contains active sites. Contains active sites. And on our illustration, you can see that these active sites are those little yellow on my screen. I know yours might not have distinct colors, but you can see in this little segment here, there's this little active site that's covered up by tropomyosin. And you can think about this as simply as sort of a molecule that keeps it capped, covers it, a cover of our active sites. And our sarcomere contraction, our sliding filament theory, relies on the idea that thick filament and thin filament must, let me put this on. Okay, and thick filament and thin filament, dang, I got hot all of a sudden, okay. And then so thick filament and thin filament here, they interact. So they interact. And so when you look at thick filament and thin filament, this relies on them to interact. And that's where we're picking up on our narrative here, is that our tropomyosin serves as a little cover for our active sites. And then troponin is sort of, bless you, is our, I'm going to call it simply, just for the sake of understanding, our gatekeeper. And I'm even going to put the key, or the password, is really going to be calcium dependent. Here, I'll write it out because I don't want to confuse you guys with ions. It's calcium dependent. And so what it relies on is that in the presence of calcium, our troponin calcium will find troponin, and then it will tell tropomyosin, hey, get off the active sites. It's time to cause our sliding filament theory it's time to contract. It's time for these two to interact in the presence of calcium. And so, now, sarcolemma is going to be our plasma membrane, as a reminder, a plasma membrane of our muscle cell. And something that we're seeing inside of our myofibril, right? Because inside of here, you can still see the position of the M line. You can see the thick filament. You can see the thin filament. And that's everything that we just discussed, all those A bands, the I bands, the Z lines, they're all underneath this structure called the triad. Ask away.
So when a muscle contracts, that is the act of calcium finding the troponin and then removing.
Yes, exactly. Calcium will trigger the interaction between thick and thin filaments. And so the nice segue into this part here, for that very reason, the triad is important, because where that calcium is stored is inside of this structure called our terminal cisternae. And if you look at this word triad, and you can see this bracket here, you can sort of see three segments. I'm going to try my best to highlight here. And you can see segment number 1 here. And you can see segment number 2 is this yellow one here on my screen. And then if I switch colors, this red one here, this is our third one. So triad implies 3, right? So you're seeing that this bracket implies those 3 segments. But if I wanted to draw more accurately, the better thing to do would actually be to pick the same color as I did for that other end on this end. Because when you look at this region, you can see that our triad is made up of 2 terminal cisternae, 2 of these. And transverse tubule, there is only 1. And maybe I should do that in green here, in green for us, just for the sake of being consistency. And so that's going to be this green one dead center here. So you have 2 terminal cisternae and 1 triad. I'm sorry, 1 transverse tubule. And so the role of the terminal cisternae is to sequester and store calcium. And the T tubules are what are going to trigger the release. So I'm going to put transverse tubule trigger release. Triggers the calcium release. Very good. Triggers calcium release. Nice. So the transverse tubule triggers the calcium release. The terminal cisternae release it and restore it when necessary. Now, this sarcolemma is a plasma membrane that can actually pass charges through it. It can actually carry charges. And so when your nerve cells decide to move a muscle, when you send the voluntary motion to control, it's connected to nervous tissue. And if we remember, when we were looking at our nervous tissue, we had what sends the passageway, the axon, and then we had our axon terminals at the end. And here we can see that they're synapsing, or they're going to connect. And we're going to see that more towards the end of the class. And they connect to the sarcolemma. And so they're going to send or relay that message downwards to release calcium from these terminal cisternae. And we'll get back to that very concept. But this is just the overall structure of how the muscle stores calcium, or where it stores it. Nothing new to say there. This is all the same thing. And then now, this structure here that you're seeing, this terminal cisternae, believe it or not, it's something that you already know. What does the word reticulum remind you of? Endoplasmic reticulum. Instead of calling it an endoplasmic reticulum, we call it a sarcoplasmic reticulum. That's it. And inside of that sarcoplasmic reticulum, inside of your terminal cisternae, you have a protein called calsequestrin, like sequester. Sequester means to gather together. So cal implies calcium, so it's like a calcium gatherer protein. And it exists inside of that sarcoplasmic reticulum to store our calcium ions. And so when that calcium ion is released, it's released into the fluid. And remember that the fluid inside of a cell is called cytosol. Cytosol. And this image, I keep forgetting. I take for like 2 semesters in a row, I keep forgetting to take this out. You can go ahead and just do this to this. Yes. Yes. And so with that, Now we're going to learn how it all comes together. And these are just the overall step names, right? Up the top, you can see the step. And then you can see the wordier explanations. That will be a nice read probably after I explain it. But I always like to explain things simply, and then it's easier to understand it simply, and then do the complex reading, I think, personally. And so if we look at our resting sarcomere, that means that our sarcomere has not contracted. If we look at our H-band, we can see it has not decreased, but by the very end, our H-band is much more smaller. So we're really going in this direction down and this direction. That's the order of these. Follow the arrows as we go along. And so here we are seeing our thin filament. And our star player, remember, being troponin, our gatekeeper that requires calcium, and tropomyosin, keeping the guard of the active sites, blocking it. And so in the presence of calcium, calcium will bind to troponin. And then what we'll see happen is that now we can see all these active sites completely exposed. And here we're even seeing our calcium bind to these troponin molecules and exposing those active sites. these active sites. And that allows now an interaction, a cross-bridging between the thick filament and the thin filament. They can now interact. So that's why we call tropomyosin and troponin together. Sometimes you'll hear it called the troponin-tropomyosin complex, because the troponin needs calcium to move the tropomyosin out of the way, to move tropomyosin out of the way of the active sites on those actin molecules. And so with that now, our myosin heads are free to interact with our thin filament. And what did the mitochondria produce for cells? ATP. And if we broke this word apart, it means adenine triphosphate. What do we think ADP means? Exactly right. Adenine diphosphate. And you can see that little plus P is that third phosphate, right? We don't really have to be chemistry experts to understand that. We have an extra phosphate. I'm just going to write P for now. And when you combined these together, circle too much. Oop. One more time. When you combine these together, a diphosphate with one more phosphate would make your triphosphate. And so what you're seeing is that when this breakdown occurs, when this phosphate is lost from ADP, it sort of allows molecules to carry out their activity. Releases energy and lets them carry out their activity. So this ATP is broken apart. It has that energy already ready to expend. That myosin head is going to bind to those active sites. The act of myosin binding to those active sites is called cross-bridging. Cross-bridging. This is a vocab word. You need to know that fact. Is that when myosin heads look for those active sites, that's referred to as cross-bridging. And so, next. Now that it used up that ADPP, right, the energy from it is sort of useless. It's like it got used up. And then we're going to need a fresh ATP molecule to come in, a fresh energy molecule to resupply the next motion. So when our myosin heads pivot, that's when we carry out, I'm going to zoom back out here, that's when we see the movement. I'm going to show you a video with animation. It looks a little bit better with a video animation. But otherwise, this occurs because that myosin was able to bind, and then it moved that thin filament towards its M line. And so our myosin head pivots, moving our thin filament towards that M line. Now, we used up our ADP and our phosphate group's energy. That breakdown, that catabolic reaction, when it broke down, it released that energy we needed, but now we got to redo it again because that thin filament's got a little bit more distance to go. And so when we see this cross bridge detaches, it's as simple as that. That cross bridge detaches, a new ATP molecule can do something that we call like recocking. Like when you think about a gun, right? And you have you've seen the cowboy movies and they go, right? On those Magnum guns, they go, That's called cocking the gun. And so it's the same thing. It's like these ATP molecules come in and then they recock the myosin, or they reactivate. They activate just like a gun. It's ready to shoot after you cock it. And so this reaction now is ready, again, going to repeat the same process we saw in the beginning. It's going to be capable of forming any more cross bridge it might need to if calcium is still present. And so that's the general, what we just described is a muscle fiber contraction cycle. Right? Pretty simple, right? The muscle fiber contracted. A contraction cycle. The muscle contracted. Now, some little more, we got to sound like college students, right? We got to be more technical. Remember that the myosin heads are energized and cocked, right? And they're only able to do so because they break down ATP. So we refer to those myosin heads as ATPase because they are proteins that utilize ATP, or they break down ATP. That's what this implies. I thought that was one of your phones. Is that a fire alarm outside? Can you make sure that back door is locked? just because I'm tired of smooth shooting now. I had to do it that like 3 weeks ago. Okay, with that. Dang, I wish I hadn't said that on the recording. Okay, anyway, so the contraction cycle now, the calcium ions, I'll have to edit that part out, I think. So contraction cycle now, this is going to involve calcium ions released from the sarcoplasmic reticulum. That calcium binds to troponin, and then it changes the position of the tropomyosin, exposing those active sites. The cross bridge can form, and cross bridge just means myosin binds to those active sites, forming that cross bridge. And then the myosin head pivots, moving that thin filament towards the M line or the center of the sarcomere. And this action is referred to as a power stroke. Make sure we know this. And then we used up the energy from our ADP-MP, right? We used our ATP up. And so when the cross bridge will detach, and a new ATP molecule will come to take its place. And now we will be able to form a brand new cross bridge because the myosin heads reactivated when they split the ATP into ADP and that extra phosphate group. That released energy recocks the myosin heads. And so as long as calcium is present and ATP is available, then we can expect that this cycle will continue to repeat itself. And I'll come back to this line in a second, action potential. And how does this all end now? We saw that at the very beginning of our narrative, we needed calcium to bind to our troponin. So the second that calcium gets sequestered by calsequestrin back into the terminal cisternae to be stored, then calcium now will undo, right, the exposure of the active sites. In other words, it'll recover it up because calcium is leaving now. It left the troponin. And so those active sites get covered again. How can you do another contraction if those active sites are covered up? And so again, we call that troponin, tropomyosin, we call it a complex because they work together even though they're not the same. They're not one thing. They're two different molecules that work together. So they're a complex. And so, to finish up, we have to go over a topic which is still built off of the same thing. Like, how did the muscle decide to release that calcium is the real question, right? Like, I hear in this segment here, in this portion here, I mentioned that the T-tubules was responsible for the release or telling, right here as we wrote here, triggers calcium release, right? Calcium is stored in our terminal cisternae, and the transverse tubule, it triggers the release. And so when you send a nervous impulse, the T-tubules will sweep through the entire muscle. And this is just one myofibril, right? We got to remember that we're looking at something like this, a bunch of myofibrils. So the second that nervous impulse, otherwise known as an action potential, simply for now, arrives like you send the voluntary command to move it. It sends and it sweeps through. And then it causes calcium to just release through the entire length of each one of those myofibrils. So the topic now is what caused the calcium release? And the answer is going to lie with the neuromuscular junction. The neuromuscular junction. This axon terminal, if we go back to our connective tissue chapter, we already learned this slightly. Let's go to our nervous tissue. Oh, wait, that's the wrong segment. No, it's not. Yes, it is. It's going to be in the epithelial chapter. One second. Oh, wrong button. Connect. No, epithelial, tissues, documents, tissues. And our nervous had the cell body, it had the dendrites, it had the axon, And I have another slide deck. I'm just going to use this one for now because I don't want to be spending too much time searching for it. But it looks something like this. And it doesn't change between textbooks. And it looks something like this. This is what your brain is essentially composed of. This is all of these forming connections, like the hardware of a computer, are what relay your thoughts, your sensations, carry out motions. So from your brain, the action or the voluntary motion to control something will be sent down this axon. And then at the very end, these axon terminals will be connected to something. And in our case, for today's topic, they are connected to the sarcolemma of a muscle cell. And so when I say nervous impulse, that's the direction of a nerve impulse. It always goes away from the cell body, traveling down its axon, and then it causes something to occur here. And that something is what this next topic is really about. And just as an FYI, I know I say the word nervous impulse, and I say it out of simplicity, but the appropriate term is an action potential. An action potential. And so I'm just going to go ahead and start. I know. And then I'm going to start subbing it out with action potential. So at the very ends of our axon, it travels to our sarcolemma, right? And then you can see that a little interaction occurs, and hence the name neuro, because the nervous tissue end here, the axon ending, combining with a muscle. Neuromuscular junction, and junction is where they are meeting here. That's where all the action is occurring, right? This is what's going to trigger at some point the calcium release that we learned about earlier. So, little details. When we look at this axon terminal is the ending of that axon terminal. The space inside, this space forms like a little cleft, a little empty space. There's like fluid in here, right? It's not like just dead space, but it's empty space because there's no sarcolemma here. There's no muscle there. It's just a space for them to communicate. And so that's referred to as the synaptic cleft. This word synaptic comes from whenever you think about these cells, whenever they're connected to anything, we say they form a synapse. And I know we haven't discussed neuro yet in our class. That's next chapter, I believe. And so synapse means when a nervous cell connects with something else, to keep it simple. And so the synaptic cleft is where it connects with the muscle. And so the motor end plate is the end or the boundary of the muscle. Nice and easy, right? Because muscle does motor movement. And when we get to nervous, something that I hope that by then you've adopted, that whenever I say the word motor, I am talking about muscle control. And we should write that down. Motor, muscle control. Motor end plate. And so you can see that it's the plate or the point or the boundary of our sarcolemma or our muscle cell. And so all together, you can see we zoom out, you would have a synaptic cleft or a synapse would be forming here. This would be the connection of the nervous cell with the muscle. And here we have our axon, and this is the pathway of our action potential going away from the cell body. And we refer to any neurons that are going to link to muscles as a motor neuron, because it's going to control a muscle. And do not forget, muscle fiber is equivalent to the word cell. Muscle cell. The word fiber is equivalent to cell. And so we're looking at one huge cell here with a bunch of multinucleated cell, and it has striations, right? And inside we saw our myofibrils, and each one of them has that repeating Z line, those sarcomeres, right? A repeating sarcomere through the entire length of it. And so that's how we get, at the end of the day, a contraction. Action potential. And let's go into more details. Okay. Now, what causes it? When we zoom in on that synaptic cleft, right, that little space here, within the axon terminal, there are vesicles that contain something we refer to as a neurotransmitter called acetylcholine. Reading for you up here. Acetylcholine. And this is considered a neurotransmitter. The motor end plate has these folds, because you can see that if you compare it to this one, you can see that the sarcolemma is very smooth here, and then you can see that there's these folds the second this begins to interact with it. So those are referred to as junctional folds. And what they have is acetylcholine receptors. And I'm just going to say ACH for short, which stands for acetylcholine. So you can see you have receptors that are receptive to acetylcholine. They bind to it. And within that space, and I'm going to use this picture now, here's our junctional folds, here is our acetylcholine receptors, and in here are the acetylcholine neurotransmitters within our vesicles. And then lastly, we have ACHE. You'll see that there's a little E here, and it stands for, as written above, esterase, acetylcholine esterase, which breaks it down. And do not forget the synaptic cleft is the space within the cell. Between the two cells. Between the two cells. Now, when the action potential arrives, finally, right, it arrives to that muscle cell, what we can expect is going to be logical here. The acetylcholine is going to look or be released to bind to its receptors. So that's what's denoted by this arrow here. And as the acetylcholine undergoes excitosis. Remember, excitosis is when vesicles release their contents into the outside space, right, outside of the cell space. And so the neurotransmitter acetylcholine is released, and then you can expect that it's going to bind to its receptors. And so when the acetylcholine binds to the acetylcholine receptor, what that allows is sodium to sweep in. Sodium just sweeps in. It's like these are proteins that require acetylcholine to activate them or open them up so that sodium can sweep through them, can go inside of the muscle cell. And now, once that sodium flows in through the sarcolemma, what it does, and I'm going to have to sort of break from this order here because I just want it to make sense now, is that sodium, once that sodium inflowed into the sarcolemma, right, goes through the sarcolemma, that's the trigger for our T tubules. That's the trigger for these tubules. Sodium. So when sodium enters that cell and sweeps through, then it causes the release of what other ion? Calcium. So that's what I meant by, they send a signal down. That signal is sodium, right? That's what happened. Acetylcholine caused sodium to sweep through the T tubules, and that alone caused the release of the calcium to finally interact with, or to cause the interaction between the thick filament and the thin filament. All right, so back to our little narrative. Now, we have to think about now, acetylcholine is responsible for sending that signal, but if you don't stop it, you can't really stop or control those muscle contractions. You could be twitching, for instance, right, if you can't stop or close those down. And that becomes a topic in physiology. And so, anything else I'm missing from there? Just want to make sure, blah, 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 raise down. All right. And so once we used up, right, like once we're done with our muscle contraction, what's going to occur is that acetylcholine esterase is going to look for our acetylcholine and break it down. You can see that it picked it up and now it broke it apart and made it dysfunctional. It made it in a form that cannot react anymore with its receptors, and then the body will find a way to recycle this neurotransmitter or the components of it to recycle it. But that's not really part of this class. But otherwise, that's the gist of what happens to those acetylcholine neurotransmitters once they already carried out their function. And now you'll notice that this arrow was the action potential. And originally it was over here, sweeping into our axon. And then it made its way, caused sodium to sort of cause this sweep. And that sodium now, you can even consider it a continuation of that signal. Like sodium, acetylcholine was released, and then it caused sodium. And then that sodium, you can consider that that's still a continuous action potential. Because if you look at it at the grand scheme of things, that when this action potential arrived, causing the neurotransmitter release, all it did was cause sodium to actually flow through the sarcolemma, and it does it through the entire length of our muscle cell. And so if we look at all our T-tubules once more, it does, sorry, let's go to this one. It's a little bit better. That it sweeps through the entire length, and as it's doing this, it's sort of like triggering each T-tubule as it's going. And so the whole muscle cell is now being triggered to release calcium through that inflow of sodium, or Na, right? Na plus sodium. And anything else I'm missing? Nope, that's it. And our action potential spreads through the motor end plate across the entire sarcolemma of our muscle cell. And that's really it. And then once it's cleared, when there's no more acetylcholine, you can expect that our action potential will not occur until it's this, you decide to move that, till the motor, till you decide to move it again. Till the nerve cell decides to move it again. And so full story now, right? But we're sort of walking backwards now. T-tubules, right? The action potential travel through the sarcolemma, causing sodium, a sweep of sodium, causing the calcium inside of our terminal cisternae to be released. And now this calcium will be free to interact with our thin filaments, troponin, to remove the tropomyosin so that we can get some cross-bridging action, right? And so altogether, this is called the excitation-contraction coupling reaction. And actually, we should need to add the word reaction here. I know that's a mouthful here. And all it means is the full story. Everything from the action potential arriving to our muscle and then causing the filaments to slide, the sliding filament theory, all together. That's what it means, the entire story. So we backtrack, I think it's the last page, really. Whoops. Oh, I just realized why that happens. Yes, that was pretty much it for this chapter. But if we go full circle and we look at the first page, here was our nervous cell, right? It sent an action potential causing excitation along the plasma membrane by the release of acetylcholine, which binds to the acetylcholine receptors. Then the acetylcholine esterase destroys those neurotransmitters or recycles them, so that way they stop triggering action potentials to travel through the sarcolemma, right, the plasma membrane. But as the action potential travels through that sarcolemma, that it travels down the T-tubules, and then what this signal is an inflow of sodium, right, down these T-tubules, causing the sarcoplasmic reticulum or the terminal cisternae of the triad to release the calcium. Calcium will bind to the thin filaments, exposing the active sites on the thin filaments by binding to troponin. With the use of ATP, you will get our sliding filament or a contraction on the sarcomere. And again, calcium finds our troponin, binds to it, allows for cross-bridging, and you can see that it causes our muscle contraction. So this is really, at a glance, everything you've learned in this chapter. And so there's just little nuances that you have to add. And then we remove this for the sake of I don't want to make you guys go crazy. I'll let physio turn you crazy there. But yes, that's good enough for us. All right, let's look up a video. I'm going to turn this off now.